So Corina is a professor at Princeton University. Among other things, she's a Kavli Frontiers of Science Fellow at the National Academy of Sciences and an Alfred P. Sloan Research Fellow. Her lab likes to study complex behavior emerging from simple interactions, and she will talk to us about the occurrence of loners in populations. Corina,
Thank you so much for the invitation, for the introduction. I apologize. Do you hear a really terrible noise, background noise on my end? Everyone can hear me fine. I have an annoying HVAC system. For the last hour, I've been trying to shut it down, and I have no idea how, but at some point, hopefully they will. But if you guys are not bothered by it, then that's perfect. Okay, so I have been extremely fond lately of this wonderful, wonderful system. This is Dictyostelium discoideum, the cellular slime molds. They have a very interesting life cycle. They are single-celled amoebae, free-living. As long as they have food, they just eat and divide as solitary cells. As soon as they starve, they undergo a very sophisticated aggregation process that leads to the formation of what at first looks like a pretty unimpressive mound of cells, but that then becomes a pretty impressive multicellular organisms with an outside-inside boundary. It looks like a slug that can crawl around on a plate. It does chemotaxis, phototaxis, and when it gets to the right conditions, it stops and it forms a fruiting body that has a stalk made out of dead cells. About 20% of the cells might die to form the stalk, and then about 80% of the cells in that original mound turn out become the spores that then are resistant to all sorts of stressors, and they can get dispersed to our other locations and restart the process all over again. Now, as you might imagine, this has been just an extraordinarily rich system for studying a lot of interesting questions. But let me first just regale you with some videos so you actually get a sense, if you haven't already seen this, of what this looks like. So this is one such collective behavior, the aggregation towards one center. It's zoomed in, so we really only see these streams. So it's pretty small scale here. That now is what starts to look to me extraordinary. So out of a mound of cells, we get the formation of a proper multicellular organism. This one starts crawling on the plate. It will quickly get out of this of the screen, of the focus, and so you won't get to see the beautiful formation of the fruiting body, but I have an even better one that shows an entire petri dish, and you get to see the formation of multiple such territories where multiple such slugs then emerge and crawl around and find the right conditions. And when they do, you see the formation of these, as you see at the bottom of the screen, the formation of these self-organized multicellular fruiting bodies with, again, 20% of the cells are dead to form the cellulose stalk, and 80% of them have turned into these resistant spores. This organism is entirely dependent on spore formation for resisting. So it's entirely dependent on multicellularity to form these spores. A single cell can't form spores. So the multicellular stage of the life cycle is essential to withstand bad conditions. So it's been a very popular model system for the last 75 or 80 years, I guess, for multicellular development, for collective behavior, for social evolution, because of this apparent, altruistic suicide that these cells perform to form the stalk. There's obviously the potential for cheating to happen, because why would you want to die in the stalk when you could be a resistant spore and move on? And so it's been a really interesting model system for a lot of exciting questions in social behavior and multicellular evolution, and more recently also in an ecological context. And I believe that they're all kind of tied together, and that's partly what this talk is going to try to link. So here's the unexpected ecological spotlight that arose over the last 10 years or so. So where do we find these slime molds? You find them in a variety of places, but I think the majority of the strains that we work with were picked up in North Carolina. So you can find them in temperate forests, living in leaf litter and layer and in the soil. They feed on bacteria, and in any kind of very small, it hasn't been done in a very systematic way, sort of a diversity survey, but every time people pick, on a small scale, try to figure out what the diversity of the coexisting strains, so that means genetic variants of the same species, Dictyostelium discoideum, what is the diversity of such strains in nature? On very, very small spatial scales, you would find pretty high diversity. Now, why is this interesting? This is interesting because this means that first of all, they're very diverse, but second of all, they are found in more or less the same place, and that means they are eating together, and if they're eating together, they're starving together. So there is an opportunity for them to undergo the whole multicellular process together with other species, together with other genotypes. So there is an opportunity for multicellular organism formation that isn't genetically homogeneous. And sure enough, that's exactly what we find in nature. Chimeras actually form readily. You can go and pick up such little fruiting bodies from nature, and some fraction of them will be chimeric. We'll have at least 2 different genetic strains in there. So there isn't really, unlike most multicellular organisms, there doesn't seem to be some way of ensuring that genetic homogeneity. They certainly don't seem to have a very strong kin recognition mechanism that would allow them to tell self from non-self. Now, as soon as people realize that chimeras form readily, then there is, of course, the possibility for even more, the possibility for cheating. If 2 strains get together, one strain could easily piggyback on the other, the formation of stalks by the other, and would just put all of its cells to the spores, and that certainly would ultimately lead to the demise of this multicellular type of behavior. And so people brought such strains, such wild type strains, into the lab. And started to form pairings, so put 50% of cells of one strain, put 50% of cells of another strain, let them allow them to be starved together on the plate, and then form the multicellular fruiting body, and then look at the spores and try to see, do we find any kind of reproductive skew? We started with 50-50 cells of each type. If there's no reproductive skew, if there's no cheating happening, then we should find 50-50 in the spores. Rampant reproductive skew, basically every single pair that was tried had some sort of reproductive skew, which suggests, rampant cheating in nature, which of course poses the problem of how does sociality persist at all, or this multicellularity persist at all, despite this apparent cheating. Even more dramatic was the finding that it's not just that you find this kind of dominance interactions, but that there is a linear dominance hierarchy, that in every pairing, strain A always had more of its own spores in the globule of spores, and so on and so forth. And so immediately this would point not just to the fact that strain A is clearly a very strong a very dominant strain, but that we should really ultimately only see strain A around. So very quickly, the question of how do you get sociality to persist in the face of this very clear vulnerability is, how do you get any kind of ecological diversity in this system, given that there is a clear winner, there is a clear dominant strain. I should say, and I should have said this earlier, maybe I'll just very quickly go back to this and play this video one more time. The way these experiments are typically done is that these cells have been grown in culture, they're still growing happily, they are taken out of culture, they are rinsed really well, so they run out, so they are completely without food, then they are plated here, and here they are starving. And as they are starving, they are starved on the plate. They are put without any food on this plate. So that's just the initial condition here, just to be clear about that. Now, as we get through this, you might ask, well, why are we just looking at the, if we really want to make this accounting very, very careful, we should count all of the cells on the plate instead of just looking at the spores, and we should do that, but the problem is it's very hard to count the dead cells in the stalk. And also, you would think they don't really matter, and if all cells aggregate, then what's in the stalk is just be what is left over from what has contributed to the spores. So in any case, this kind of reasoning ultimately led to everyone focusing primarily on the spores and counting those carefully, and then reaching these sorts of conclusions. So. How I got really interested in this is that as I was at a conference and I was listening to someone talking about this and showing these beautiful transfixing videos as far as I was concerned, I noticed the following behavior. And this is a monoculture. This is just one genetic strain here. There isn't anything else going on. But notice that as all cells aggregate towards the left-hand side of the screen, and they already did the aggregation process, maybe I'll play it one more time, there are some cells that just don't seem to react to this whole massive synchronization process that is occurring. All of those cells that are towards the upper right corner, and then you see them all over the place, the plate, they simply do not seem to respond to the aggregation process. They remain behind, and sure enough, they never get to be part of this aggregate. They also eventually die because, as I mentioned, this species cannot form spores as single cells. So these cells are doomed to die. But the interesting feature about them is that they're actually pretty long-lived. They live for about 200 hours. And in 200 hours, if you live in an environment that is the forest floor, which is a pretty unpredictable environment in terms of where the resources might come next, there might be some opportunities for resources to return. If resources return, then there might be some possibility that these cells could start to eat and actually do something interesting. So that's how I got excited about these cells, that I thought maybe they are some sort of bet hedging strategy. Now, sure enough, the reason I got excited about that was that as soon as you account for such cells, if your accounting is not just spores and stalk, but it's also something that is left behind, then it's not sufficient to count the spores. Counting the spores doesn't actually tell you everything about what's happening on that plate. And you could get reproductive skew in the way that I'm showing here, reproductive skew between red and blue, even though we started with the same number of cells for these two genotypes. You could get it simply because they might leave different numbers of cells behind. But this would only be evolutionarily meaningful or meaningful in general if these non-aggregators, if these loners that are left behind are actually part of the strategy, the life history strategy of this organism, rather than just some sort of random byproduct of an aggregation process that might be too complicated, might just every once in a while have some stragglers that aren't able to make it into the full aggregate. So I got very excited about those cells. I got excited because that would mean that maybe things wouldn't be as paradoxical as we thought. Maybe there wasn't a linear hierarchy to begin with. Maybe there would be more nuance to how we should then peel layers and layers in these reproductive skew results. I also got excited because I thought they could have some sort of interesting ecological meaning to them as kind of a bet hedging strategy. They would be the individuals that are left behind to take advantage of what resources might come back, and I'll get to that in a second. But the major question, as I set up to try to think about this was, of course, are they just accidental and undesirable and maybe even unviable? Maybe there's something wrong with them. Maybe they can't sense the signal, the cyclic AMP signal that the others emit in order to coordinate. So are they broken somehow? Or maybe even if they're not broken, are they somehow accidental? You can't coordinate millions of cells without maybe missing some here and there. Those two possibilities had to be kind of dealt with, and then see if there's really some potential to these loners. The first part, the unviability, turned out to be actually pretty straightforward. So that was the first thing that we did here. And this was a collaboration empirically with Alison Sgrove, who was a postdoc here at the time in Thomas Gregor's lab, one of my colleagues here in physics. And she just set out to do this very simple experiment. So in panel A, we started with a bunch of starved cells, as one always does. By panel B, they have formed their fruiting bodies. In panel C, Alison goes in very carefully and picks up either the entire fruiting body or at least the globule of spores. So she picks up everything that is she picks up everything that is spore, so that what we have behind are either dead stalk cells, and we know that those don't do anything interesting, or the loners. And then she puts a layer of food on the plate in panel D, and then we wait. And here what we want to understand is, are those loners able to eat this food? Are their progeny, ultimately, when they starve, able to recapitulate the self-organization process, the multicellular process, or are they broken somehow and they just never get there? Sure enough, after about 72 hours, I think, she came back and she found plenty of fruiting bodies on this plate. So that meant that the loners were able to eat, to divide. Their progeny were perfectly able to recapitulate the self-organization process, and they also left their own loners behind. So this was promising. And I mentioned, and so at that stage, that was the best that we could do, just convince ourselves that they were at least not broken somehow. And then we went for, we tried to complete the picture of why might they make sense as a life history strategy. Well, aggregation turns out to be costly. Cells in the aggregate, it's not just costly because you have a 1 in 5 chance to die, but you also, all of the aggregation process past a certain stage is committed, and the aggregation lasts for about 20 hours. So if food comes back in the environment, you can't stop the process, revert to being single-celled, and then proceeding to eat and to divide. You have to continue with the process, carry it through the end, make the spores have to sense the food, and then they have to germinate, which takes about 4 hours, and then they can start eating. Now, if food replenishes in the environment, any cell that is just hanging around there and is not part of this whole process can exploit it and divide every approximately 4 hours. So it is pretty costly to be part of that aggregate, even if you didn't have the pretty high chance of being one of the stalk cells and dying. And so we thought, you might be in a situation, the hypothesis was that we were working with was that loners are kind of like an exploitation strategy. If you live in an environment that is sufficiently unpredictable, in a stochastic environment that is sufficiently unpredictable, then it might pay off to leave behind some loners that could eat the food whenever it comes back, but also invest some in the spores, because that's your survival and exploration strategy via dispersal. And then the idea would be that coexistence could be ensured by a moderate dispersal between patches of the forest that might have conditions that are a little bit better on average versus conditions that are a little bit worse on average. So this was a simple model that we made. Here on the y-axis is the investment in aggregation, basically in spores, or the whole aggregation process. 1 means that everything aggregates. There are no loners. 0 means nothing aggregates. Everything is a loner. On the x-axis, we are varying the environment, and this is an exponentially distributed next food pulse. So the next food pulse is drawn from an exponential distribution with the average starvation time given there on the x-axis. So on the left-hand side are pretty safe environments where food comes back very quickly. On the right-hand side are very harsh environments where food basically takes forever to come back. And sure enough, if there's no dispersal, so you can think of each one of those columns as kind of like a patch that we are going to connect ultimately via dispersal. If there's no dispersal between these patches, then there's going to be only one winning strategy in each one of these patches. If you have very harsh environments, you should put everything into aggregation. If you have very friendly environments, then you should put everything into then you should just leave everyone behind as loners. There's no point to make an aggregation process, to go through an aggregation process. And in between, you're going to find some mix between some fraction that goes to aggregation and some fraction that stays behind. So that's the bet hedging part. The interesting part was that as we add a little bit of dispersal, so now you can think dispersal fraction of 1% means that I can now think about these columns as kind of as basically islands, each one with its own conditions. And every time spores are being produced, 99% of them remain on the parent island and 1% get distributed equally across all the remaining islands, or all of the islands. So now there's some possibility that cells from one environment might end up in other environments. As soon as that happens, you see that the diagonal starts to spread out, and so in all directions. And so you start to see that in a given environment, you might have several different strains that can coexist. As you increase the dispersal fraction, you further increase this coexistence. And you get, at about 20% dispersal fraction, you see that you get all sorts of a diversity of strains coexisting in every single environment. The interesting thing is that if your dispersal gets too high, then what you get is more or less one super bet hedger strategy that basically sees all environments all the time, almost equally likely, and optimizes for all of them. So eventually you lose this coexistence at very high dispersal rates, but at small to moderate dispersal rates, you can maintain coexistence in all of these different patches just out of their different local investments in spores versus loners. So we had a proof of principle. The loners were unviable, were viable, so we were pretty happy about that result. And so so if only we could now just prove that not only are they viable, but they are a meaningful life history strategy rather than some random byproducts, then in principle that could deal with both of these issues simultaneously and rephrase the problem. Also, it makes this, or it would make this organism more interesting because instead of thinking of it as kind of a one-dimensional fitness, it's just your investment in spores. Now you have these two different things that can give you, that contribute to your ultimate fitness, both the loners and the spore production. So it would change a little bit how we even pose the questions about cheating and all of that. But where are we on this particular question? This turned out to be extremely hard to do, and I was very lucky that at the same time that I was kind of realizing that this is a hard problem, I had a phenomenal graduate student apply to work with me, Fernando Rosina, who is now a postdoc at Harvard Medical School in Michael Bain's lab. And he was just really interested in asking these questions theoretically, but was game to try for the first time to also tinker a little bit in the lab and just kind of familiarize himself with the with this particular organism. He turned out to be really just extraordinary, both theoretically and experimentally, because here was the difficulty. The difficulty was that this is sort of an exaggerated, every single loner on the plate is represented by a gray dot that is a little bit bigger, so you can see that they are pretty much all over the place. I'm going to zoom in now on that little red square. So we would need to count these loners in order to be able to do repeated experiments and then try to figure out whether their numbers change according to some pattern or whether there's pure randomness all over the place. But as we look at that particular square, this is what it looks as you zoom in. Amoebae are not pretty cells to count. They are not rod-shaped, they are not circular, they are amoebae. So they are, you can find them in all sorts of shapes. And it's pretty hard to tell whenever you see one of these messy areas there, whether you're seeing one cell, multiple cells, how many multiple cells. So it turned out to be really hard to try to count these until Fernando had just an extraordinary insight that all cells, when they get stressed, they become circular, apparently. And so he very gently sprayed with a handheld humidifier, sprayed sugar water on top of these loners, stressed them, and the next thing we saw were beautiful spherical loners. And these you could count, and you could count perfectly. I mean, he could count them within, just he eventually developed computer algorithms to do that for the many pictures that he had to count, but the error rates were negligible. So once we had that, we could really ask, what we wanted to see was, what would happen to this picture? As we increase on the x-axis, we see the plated cell density. So we start with cells that are, that are with no food. They're starved on this plate, but we start with different initial plated cell densities, and we want to look at the loner density. At the beginning, we would expect that all cells will stay as loners because their aggregation process is a density-dependent process. So if you have too few, if your density is too low, you're not going to be able to initiate the aggregation process. So we expected that there would be a linear relationship between up to a certain density between loners and plated cell density. They would just be one-to-one. Past that density, our hope was that we wouldn't see just a mess, that we would see some pattern, right? Sure enough, as he did the experiments, and my hypothesis, the hypothesis on which we had built the previous results with the bit hedging that I just described, was that there would be some sort of stochastic switch, basically the easiest kind of thing that seems to be happening a lot in biological systems, that these loner cells would flip a coin, and based on that, they would stay behind as loners, or that cells would flip a coin, they would stay behind the loner with a certain probability, or leave with a certain probability. And so therefore we would expect a linear relationship in between the loner density and the plated cell density. As Fernando did the experiments, he saw that sure enough, he confirmed that first portion of the plot. That was not the most exciting. But then what he found was definitely not a linear relation, definitely not a one-to-one relationship between loner density and plated cell density. Instead, he found a constant number. Loners seemed to be basically the same number of loners seem to be left behind basically past a certain density no matter how many cells we started out with. And that was pretty surprising because I don't think I had ever worked on I had ever even thought about theoretical models that might explain how a system might count a given number rather than produce a given conserved fraction. So anyway, the good news about this was that they certainly weren't random. In fact, it was a pretty clear pattern that we were observing. So all of a sudden, there was something that was clearly worth investigating. They're definitely not a stochastic switch, but they seem to be part of somehow the life history strategy of this environment, of this organism. And we can look at different genotypes. All of these genotypes are wild isolates, so their behavior hasn't been transformed in any way in the lab. And we can see that they have different numbers of such loners, which is why we ended up declaring that this was a heritable strategies. So red and blue here have significantly different numbers of loners. What we are plotting here is the number after that initial stage. So it's the constant number and then the error bars. So the other thing he did, and this was almost accidental, how he realized this, it was to try to was to see that these numbers were very heavily influenced by the porosity of the agar. And so at more porous agar, there were far fewer loners. All strains had fewer loners. At less porous agar, they had more loners. But notice also that the differences between strains aren't proportional as you go across these different levels of agar concentration. They're clearly more porous agar makes all strains more similar, less porous makes them even more different. So it seemed that loners were a heritable strategy that was also influenced by abiotic factors. And one natural reaction would be, well, you know, porosity of the agar is a very it's like an artificial environmental or abiotic factor because it's something that pertains to the lab. But because these amoebae live in the soil, the porosity of the soil surely is an important element of their usual environment. And so we could think of this, perhaps imperfect, but we could think of agar concentration as a proxy for something that they would encounter as they live in their natural conditions. So where to take it from here? What kind of dynamics can account for this behavior? Well, the typical models that were used to model anything about the self-organization of Dictyostelium discoideum, there's been lots of modeling done of that, very elegant models. Here's one from Kessler and Levine from the 1990s. It's primarily focused on trying to understand how these streaming aggregates form. And it has many features. I'll just reveal all of them. The point isn't to go through this in particular, but there are models that can reproduce this very sophisticated behavior of coordination and forming of these streaming streamers, these streaming cells, and their aggregation towards the center. And they require various elements, many of which have been tested experimentally. We know that what they need is cyclic AMP. We know that there is some threshold for the activation of each cell. We know that there is some sort of relay mechanism that gets cells to emit a pulse and create this pulse cascade. We know that there is some sort of recovery time. And we know that there is some sort of gradient that is being established that leads to a directed movement towards the center of aggregation. So very elegant models, but none of these models create loners that have this sort of behavior. None of them even asked anything about loners, because within these self-organization models, the target was to think about the coordination movement and not so much about any cells that might be randomly left behind. But we had reason to suspect, and I'll get to why towards the end, we had reason to suspect that cyclic AMP was not the molecule, or I can actually say it also already. I revealed those results with the porosity of the agar and how sensitive the loner numbers seem to be to the porosity of the agar. It turns out that the differences in agar that I was showing there, and I was just saying more porous, less porous, those were 1% differences in agar concentration. Cyclic AMP is a very small molecule that should not be influenced by, should not be sensitive to such small differences in agar concentration. So we immediately had a suspicion when we realized that when we got the results about the agar concentration, we immediately had a suspicion that it's not cyclic AMP that is really driving the partitioning of the population into loners and aggregators, that there are other larger molecules. And such larger molecules have been described and typically implicated in earlier stages of the process, not in the aggregation stage. So we already thought that we probably need to come up with a different way of thinking about them and a different kind of model. And so we vastly simplified the aggregation part of the story, and we focused more on the pre-starvation stage, where where other molecules might interfere and might lead to some partitioning. So here, this middle panel, the aggregating cells, is basically a caricature. As far as anyone who studies the coordination with the cyclic AMP, as far as anyone who studies that is concerned, this is just a caricature. We're saying there's going to be a center and cells are going to move in that direction with some strain-specific velocity v, and we're not going to model anything of the bells and whistles of that coordination, of all of the streaming and all of that. What instead we're going to do is we're going to add a state prior to that, and that's the pre-aggregating cells, and we are going to actually primarily focus our modeling and our thinking on that state. And in that state, we're also going to make all sorts of simplifying as well. We're going to assume that there are no resources, because that's how usually these experiments start anyway. But we're also, and therefore no division, but we're also going to assume no motion of any sorts, and we're going to assume that cells emit a signal that is a strain-specific signal. It's a generic signal. All strains can sense that signal, but the rate at which it is being emitted is strain-specific, and that rate is gamma. They're going to perceive the signal with a strain-specific sensitivity threshold theta. And then that signal diffuses away. It's the same signal, so this is not strain specific. It has a certain diffusivity constant d and decays at rate eta. So that's what we assume, that they emit a signal in this pre-starvation state, and that they have sensitivity to that signal, and that both of these are strain dependent. And then we further assume, and this is the major departure from previous models, we further assume that when your threshold has been met, when the signal concentration that you perceive exceeds your threshold, the decision to aggregate is not deterministic, it's stochastic. So it's a quorum-based transition rate that is stochastic and strain specific, and happens at a strain specific lambda. So once your threshold has been met, you are not automatically going to, but you have a certain probability per unit time of making the decision to start the aggregation process and be moved in that other box. Once you're in that box, you start moving in the direction of the center, you move with a certain strain-specific velocity, but you continue to emit that signal that the pre-aggregating cells are also emitting. So this would be in addition to your cyclic AMP. And then eventually you get multicellular organism. Now, this is how the model would look. So this is a plate here that has high density. All cells have quorum. All cells are in blue. That means that all of their thresholds have been surpassed. The aggregation process starts as they decide to aggregate. They turn yellow and they start moving in the direction of the upper right corner. But as these cells go away, slowly by slowly, any cells that used to have their quorum met but didn't make probabilistically did not make the decision to aggregate and switch their state are now left behind with not enough neighbors to have that quorum met again, and so to continue to have that quorum met. So basically, our hypothesis is that the loners are these P cells, that the cells that used to have a quorum, but they just didn't make the decision to aggregate at the right time, and they're probabilistically, and then they were left behind, and now they don't have quorum met anymore. This is a simple hand schematic, but the model results then predict exactly the behavior that we see in the experiments. They predict that initially there's not enough quorum, there's no aggregation. Eventually aggregation proceeds, but too few cells begin to aggregate because they're still too sparse, and so there's some intermediate inefficient area. And then eventually there's a plateau, and the cells that are left behind are P cells that lack quorum. And we did this both simulations that were spatially explicit, but we also were able to do a spatially implicit calculation in the limit of high diffusion rate. We can actually prove that such a plateau exists, and we can calculate that plateau. So we can calculate the number of loners, and we can, it looks like this. Again, theta is the strain-specific threshold, gamma is the rate at which the signal is being produced, lambda is the rate at which they make the decision to, once their quorum is met, the decision to aggregate, and v is the rate at which is their speed. Velocity. Just to give you a little bit of a sense, I mean, you can see there how they depend on the various different parameters, but to give you a little bit of an intuition for this model and for what changes the number of loners that we see behind, let me focus on, velocity for a second. Velocity is very important because the higher the velocity, the faster the aggregating cells get away from where they used to be. So the faster they get away and they stop emitting, and therefore their contribution to the signal around the remaining cells is diminished faster. And so the faster the velocity of the aggregating cells, the more loners we should see behind. On the other hand, the slower those cells, aggregating cells, are getting away, the longer time they persist wherever they all are and continue to emit signal and therefore maintain quorum for everyone else, so that the others have more time to keep flipping the coin and making the decision to aggregate themselves. And one can go through all of the other parameters, and there's that big expression there. Notice also the dependence on the diffusivity constant. So the prediction here is that the higher this is from the spatially explicit version, the higher the diffusivity constant, the fewer loners you should have, and then the lower the diffusivity constant, the more loners. And the idea would be that the better the signal is that's spreading and diffusing around on the plate, the easier it will be for cells to sense that there are other cells still left around, even when they are less densely packed. And so they have more time to make that decision. So, not only that, but we also notice that as we vary the signal diffusion coefficient, and so here the arrow goes from right to left, just to match the agar concentration. So as the signal diffusion increases, as the porosity increases and as the agar concentration decreases. It's a little bit, I'm sorry, this axis is always kind of a pain to explain. But basically, the more porous the agar, the higher the diffusion that you would expect. Notice that as we plotted our results, we are also obtaining, as a function of the signal diffusion coefficient, we're obtaining not just the prediction that that you should have fewer loners when you have higher diffusion, but also that the differences between strains should be exacerbated at lower diffusion. So we were kind of recapturing the results from the agar concentration experimental findings. So this suggested to us that maybe, the diffusion of the signal might indeed be what is behind these loners, or might play a really important role. And in order to test that, and again, because this is the agar concentration here changes from 2% to 3% to 4%, and these strains seem to be, these loners seem to be sensitive to that, that really tells us if the theory, if we believe the theoretical results and diffusion coefficient is indeed what's behind this discrepancy there, then that really tells us that it's not cyclic AMP. Cyclic AMP is too small to be sensitive to such small differences in agar concentration, and that it has to be one of these larger molecules like condition medium factor or pre-starvation factor. So it gives us a candidate for how to continue to look at what determines this loner behavior. So in order to do a more conclusive test of that, then Fernando set up these really neat experiments. The control is simply the comparison that I showed you before. It's the same red and blue strains in the same conditions. The water film is the same way of putting cells on a plate without any more food. So the same process is in control, but adding a thin layer of water on top of them that would dry up before the actual aggregation process happens, but would in between those two endpoints, like between the aggregation process starts, in those 4 hours or so that it lasts on the plate, it would mediate the diffusion. It would make it easier for other molecules to diffuse. And sure enough, the prediction was that if we have that water film, we should see fewer loners, and that's what we saw. The other is another cool experiment that he did that where the conditions are actually quite different. Now we're trying to starve them in a flask where we're trying to make the signaling as well mixed as possible. So a different kind of initial condition. So instead of growing them exponentially in that flask and then stopping at some point, taking them out, washing them and putting them on the plate to starve, we actually continue to grow them in that rotating flask, let's say, until or mixing flask until they starve right there all together, and then they have plenty of time to mix all of their molecules for everything to communicate with everything else. And so the prediction was that should also decrease the number of loners. But because we see a reduction in the number of loners, even in that experiment, even before we even put them on the plate, after we put them on the plate, the conditions are the same as in the control. All we change is what happens to them as they grow without any kind of spatial structure. That really convincingly shows that the partitioning behavior in this population happens very early. As soon as they sense the very first stages of starvation, before any of the spatial structure and anything else, there's already a beginning of a process of partitioning, which we thought was interesting and exciting. So we could, with two states, pre-aggregating and aggregating, and the stochastic quorum-based transition, we could recapitulate qualitatively the observed behaviors, including the constant number of loners left behind. Because in real life they're also using the same signal, we expected that means that they're also probably very sensitive to biotic interactions. I won't go into this part of the story, but this is also really interesting. So we made mixes of different frequencies of the different initial frequencies of the strains, and we could see that the loners were impacted by who was present and not in a linear combination kind of fashion. And that has all sorts of ecological impacts for the diversity. So I'll end with saying that basically, ever since I started giving this talk, but also ever since I started doing this work and talked to other colleagues, almost every system in which people have thought about collective behavior seems to also exhibit some sort of loner behavior. Whether we are talking about the great wildebeest migration, it turns out that in this system, 1.2 million wildebeest will go and do the whole migration process, but about 200,000 of them stay behind in residence. And they are not the same 200,000 or offspring of the same 200,000 from several years ago. They're just different individuals seem to be taking on that role in different years. And plenty other systems, certainly lots of microbial systems. Locust swarming seems to have its own loners. There are at various pockets or areas of the gigantic locust swarm, locusts peel off and they revert to being solitary, friendly grasshoppers. Salmon migration has this. Human systems, one could argue that there are all of these loner behaviors. None of these systems have been really amenable for asking questions about these loners and the mechanisms, and whether they are meaningful in some way or whether they are just some sort of random byproducts. But I think Dictyostelium discoideum opened up this really interesting opportunity that loners could actually be meaningful, and that when we look at collective organization in systems, we tend to focus on the extraordinary aspects of the collective process, but overlook what might be other strategies, and that those other strategies might actually be extremely important for this robustness of the system as a whole. So we're trying to look more broadly at whether this lack of synchronization that we might see across, that might exist, could actually increase the robustness of the system. How would this work in slime molds? Well, I get back to the cheaters dilemma. So we do know that cheaters exist in slime molds, and so loners are a really interesting strategy, not just as a bet hedge, not just as an ecological bet hedging, but also sort of an evolutionary bet hedging. If you're going to create a multicellular aggregate that is vulnerable to cheaters, very vulnerable to cheaters, then leaving behind some cells that aren't part of the aggregate means that those cells aren't actually vulnerable to the same things that the aggregate is vulnerable, and their offspring can recapitulate the multicellular behavior when they themselves starve. So in some sense, it's kind of like an insurance strategy for the multicellular behavior if you don't have other mechanisms for protecting the any particular multicellular aggregate. So we've been recently looking at some of these other systems and trying to understand what mechanisms. The mechanisms are probably very different for all of these different systems, but nonetheless, the loners might actually end up being meaningful in a lot more of them than we would have believed maybe before this work on butyricilium. So with that, I want to thank my funding sources and my collaborators, and I am very happy to take any questions if there's still time, which there is, I think.
Thank you so much, Corina. Thanks for the great talk. Maybe for questions, we first go to the room in Augusta. Matthias, are there any questions in the physical room?
Yes, we have some questions.
Hi, Corina. Thank you for a wonderful talk. I'm interested in this last slide about how you think about loner behavior in other complex systems. I know that you've done some work on information cascades on social media and polarization and these kinds of things. Do you see any analogies between loner behavior and what you studied here, and maybe alternative ways of participating in these kinds of opinion dynamics kinds of systems? Do you see any resilience in those networks that gets provided by loners?
It's a very interesting question. We only very briefly looked a little bit at loner behaviors in human systems. I have to say that this working on human behavior is something that's a very recent shift for my lab, and I'm still kind of feeling my way around all of the literature and the many complicated and messy dynamics of human behavior. But I do think that there is some interesting potential for study for thinking about. I mean, loners had already the idea of loners or individuals that sit outside of the game for some time had already been proposed maybe 10 years ago or more now. It was mainly, I think they were mainly called volunteers in those, but like individuals that just choose to sit out of the game for some, that's just part of the strategy. You don't have to employ that strategy forever, but every once in a while you might just kind of sit out the what would otherwise be like a public goods game or a prisoner's dilemma game. Those kind of loners could create these rock, paper, scissors kind of dynamics, these cyclical dynamics. And as soon as defectors would be able to start to like grow to really big, to really large fractions of the population, loners would start to do really well because they weren't playing the game, which meant they weren't getting much benefit from not playing the game. But if you just are surrounded by defectors, you're actually better off not playing the game. And so this idea, I think, has already been around in human systems, but not so much in this context of the political polarization and the information. So we are thinking about how to include it there. Mostly we've just been focusing on these other systems. We actually are trying to wrap up a project on the wildebeest, where I think there is a real interesting source for why there are loners there, and they seem to also have the same like constant number of loners and whatnot. But yeah, it's a good question.
Wonderful, thank you.
Hi, so thank you for the talk. It was very nice. I was wondering about how do how do they actually choose where to aggregate to? Like, what makes them realize what the center is, and also if there are a few aggregation happening at the same time, what would make them choose where to go?
Yeah, great question. So this was work that my colleague and collaborator Thomas Berger did. I think in 2005 he had a paper out in Science showing that, and this is another thing that these systems are intriguing, because there's always the question of, are there leaders, are there followers, right? Are there some cells that are going to be leaders always? Turns out that there is nothing about a center that is a leader, that makes it a leader. It's just that all cells in that pre-starvation state where they get really jittery will have some threshold for how much they can stand it, before they start to scream for everyone else to aggregate, So stochastically, at some point, you're going to get some are more, they're going to just say it, we're starving faster than other cells. So he showed it's a very much a stochastic process to determine who the center is. And once the center is determined, all of this, relaying and whatever creates the gradient, and they go in that direction. How do they choose between different centers? It's the distance. They usually choose, they choose the one, it's kind of a competition between the centers. And the cells that are closer to one center will go in that direction, and the cells that are closer to the other will go in the other direction. And naturally, as you might expect, loners end up, more loners end up being on the borders of such territories than close to a center. They're going to primarily be away from centers, the loners. That's what makes it, yeah, but that's the question.
Do they know the distance from the center? Or,
I think they just, I'm sorry. I just wanted to show you, you might even be able to see it in this video. It's the strength, I'm sure it's the strength of the gradient that I think my colleague, Tom, this is embarrassing. His last name is Cox, did a really beautiful work on showing how basically as these territories form, there are waves of cyclic AMP that are created from the centers of each territory, and then there's crashing. Those waves crash into each other and mostly kind of annihilate each other at the borders between the territories, and I think that creates a directionality. So he has done work on where do you go, where do you choose to go.
okay, thank you.
Mm-hmm.
Hi again. Yes, I actually talk very much too.
Thank you.
Thank you. The main question I wanted to ask was already just asked. Maybe I could elaborate on this a little bit. If I understood correctly, do you say that the center or the eventual center is somebody who itself cries out the loudest, or?
Mm-hmm. Not the loudest, but the fastest. It's the first one that can't take it anymore and just says, we're starving, more or less. That's how that very, simple way of thinking about it, for sure. But yes, it's just like stochastic, like the thresholds of these individuals probably have like minute differences, or there's like some stochasticity, and which one just decides to say it first. Yeah.
Do you think there is a chain reaction so that the cry of that individual does not possibly reach very far, but because it wakes up some others, then they wake up?
Absolutely, and they are all primed for that. And I'm trying to see if I still have this. There's a wonderful website. Let me just stop share and I'll reshare so I can show you this extraordinary. It's a very cool website. I just waste in. I spent inordinate amounts of time playing with these simulations, but this is a simulation of this 1993 paper. This is, let me reset it. These are all the cells. A center has just flipped its coin, it's screaming now, and the others, they're all primed to hear that screaming because they've all been starving there for a long time. And this is what happens. This is the, and I think I made a super diffusive cyclic AMP here. You can play with the, but this is what happens, right? So there's a pulse that's being emitted, and then the other cells take it in, and they relay it to the cells next to them. So it's a relay mechanism. But the pulsation is very important to establish the gradient. And so what is required here is not just the relay, but also recovery time. As soon as a cell has screamed or has emitted its signal, it stops for a second. And that's what's called the recovery time. If you don't have that, and I restart the same, oh, sorry, this is if I don't have that and I restart the whole thing without a recovery time, sorry, from the beginning, they don't do anything except locally. They can't create the relay waves. They can't create the gradient towards the center. So it's a really fascinating process. And most of these things have actually been teased experimentally also. Now we know that this is pretty much how it works.
So you think if the message arrives once or like continuously, it does not cause the action?
Yeah. Exactly. So you have to like say it, pass it on, but not just keep screaming it as you pass it on, but stop, and then the next one says it, the next one says it, and that gives the directionality of where is it coming from. Because if I just scream, how do I, so there's a little bit of as soon as I turn this off, maybe I'll make this a little bit less insane. It can create these pulsating waves, and the pulsating waves tell you, don't go that way, go this way, even though now everyone around you is kind of activated.
Oh, it is in the direction.
Yes, exactly. Because once everyone becomes activated, then you won't know which way is it coming from, right? So you need to know to stop and then, yeah. You need to have a direction in which you pass it on, basically.
I see. Another question, somewhat related to the direction. Once there is this multi-cell creature, at least in the simulation, it looked like most of them were going in the same direction, like up the Petri plate and how would you explain the directionality? Or let's say, which direction will the creature go? If this is something local, then why would they all go to the northwest, like in that case, or most of them?
I think it's this whole. Sorry, now that I switched, but I should have stayed on the previous. It's the relay mechanism. So it's this first one emits the signal, activates everyone around. That first red dot there activates everyone around. They get activated. They relay it. The others relay it, and so on and so forth. And then the center keeps emitting, right?
Of one of the creatures, right? So there are several creatures forming.
Yeah.
And it turned out, at least in the simulation you showed before, that almost every one of those creatures separately went like up and left somewhat. Upward and left.
You mean this one, in this simulation?
No, in the one that you showed before, in your own...
Okay.
This may be showing one creature, but when you show the like creatures walking, right?
I see. You mean after they become multicellular. Sorry, everyone is going to get whiplash from all of my switching around. I was on the right video after all. Hold on. Let me just playlist then. Yeah, so you mean after this process has already happened, where do they all go? Oh, that's such a fascinating question. They're in principle on this plate, they sense all the conditions are more or less the same everywhere. There's just no food anywhere. People have done experiments where you give them, so these slugs can do phototaxis, they can do chemotaxis. If you create a gradient for them, they will follow it. So if you make, like, you put them in the dark, you create a little bit of like light or photostimulation somewhere, they'll just follow that path. Here, I think they're mainly random walkers, but one thing that they don't like is to run into each other, so they will avoid each other. And even when they form, like at the bottom of this picture here, they form these two stalks. If the two stalks happen to form close to each other, they are going to try, like they're going to be leaning a little bit away from each other. They emit some sort of, I think it's ammonia or something like that, that makes them kind of separate from each other. Yeah.
And if I may, do I have time for a third question?
I don't know. The organizers are in charge. I have time. Sure, of course.
Okay, sure. So thanks. So it's about the loneliness.
Yeah.
You said that you suspect that there is some kind of evolutionary benefit. Now, if I understand correctly, evolutionary benefit should be a benefit for the specific individual and not for the species, right?
Absolutely. Oh, yeah. Mm-hmm.
So the loner itself should have some, I mean, if you're saying something like, suppose the aggregation fails, I don't know, the slugs go somewhere and die there, and so this was not useful. But let's say if this is a small probability, then for each particular loner, that loner seems to be choosing the small probability or the small expected value.
Oh, no, that's a very interesting point. I think it's, and I have to say, the evolutionary modeling of this, we haven't yet done. I'm still thinking, we're still thinking about the best way to approach it. That's why I left it at the end as more just kind of an intuitive sense, but we haven't proved any of this. But what we have proved is that ecologically, it will make sense to leave these loners. These loners will get an immediate benefit on average, just from the uncertainty of the environments that they live in, because they'll be able to just eat. If food comes back to the environment, they get a head start in eating that food relative to anyone else, including the spores. So on short time scales, ecologically, they could simply be a smart bed hedging strategy. But it could also be that in the really long term, that might have, that ecological opportunity might also have provided this interesting evolutionary insurance strategy. You see what I mean?
Okay, I don't see.
You sorry?
I don't see the benefit for the single one, but I don't know. Okay.
Well, we don't think of the loners as a single individual, right? They're
Also, the loner does not have offspring, right? So
of course, if it eats, it will make offspring, right? So I think, if we think about the genotype, right? It's the genes that are, it's the genotype, that genome that will get selected on, that partitions the behavior into loners. It's not that a loner will forever be a loner and its offspring are forever going to be loners. It's just that it happens right now that this one stayed as a loner. Some became spores of a particular genotype. And both of these strategies contribute to a fitness of that genotype, of that strategy, right? So this is not a species level argument, but simply a genetic, like this is the one genotype that decides to partition the behavior in this way versus another genotype that might partition it in a different way. So loners aren't forever loners. They're not mutants. They're not aberrant in any way. They will eat, they will divide, their offspring are going to create multicellular organisms also, and so on and so forth. And so there's no, it's not like a separate, species or genetic type, yeah.
Okay, thank you.
Hi again. I just wanted to ask another question about how the loner amount is actually constant, right? Correctly, it's because they have some kind of velocity that allows them to come to the center, right? And then loners would just stay. So did I understand correctly?
Yeah, so this is, the schematic, right? So everyone starts being, triggered and primed and so on, but they flip coins, right? Every time step, they have a certain probability of deciding, even though they're all ready to start aggregating, they're not making, this was the stochastic quorum-based decision from this previous slide, that arrow. And then the idea is that as some of them make that decision earlier, probabilistically, and they start to move and they move away, as layers and layers of them peel off and they start moving in the direction of the center, eventually some of these cells that used to be blue, that means they could have turned into yellow had they just flipped the coin right, they are left behind, but they're not blue anymore because they don't have enough neighbors that would tell them, that would meet their quorum, right? So they are just, how should I say, they're kind of like laggards. They are the ones that just didn't make the decision at the right time, even though at the beginning they were the same as everyone else. And probabilistically, any one of these, I guess, you could argue, could have done that. So why this ends up being constant, I'm going to have to tell you that this is a huge source of frustration for me. The only reason I actually believed that this was constant is that I ended up calculating it. And until I made this proof that it has to plateau there, I thought, well, who knows, maybe like in the really long term, maybe they will, like these tails will taper off. But no, you can actually prove mathematically that the system has a plateau, it has a limit. It's just that I can't intuitively tell you why all of these ingredients, I can tell you what accounts for the loners. I can't say that I have an intuition behind, that tells me why exactly they are perfectly constant. I need to understand this better, but I've proved it, so I'm perfectly comfortable with saying that it is. I see a hand up there, or Manish.
I was just.
I'm sorry. Or is there someone still in the room?
I was just wanting to ask if you try to lower the velocity of the genotype and maybe it would be linear if you lower the velocity.
That's what these, the velocity terms are all of them appear here, right? So if the velocity is higher, we have you're going to have more loners, and if the velocity is lower, you're going to have fewer loners. But there is also really important in the analytical calculation, the relative magnitude of the stochastic rate versus the rate at which you move away, basically the velocity. Their relative sizes matter for whether you're going to have loners or not. If these cells here, the yellow cells that are starting to aggregate, move very, very slowly, much, much slower than the stochastic the rate of stochastic decision making, you are not going to have any loners. So it's the relative rate of these two arrows, basically, that is very important for whether you have loners at all. If lambda is greater than V, then you're going to have loners. If lambda is less than V, the prediction is that you should not expect any loners, because basically the aggregators move so slowly that everyone will have time to flip the coin the right way and leave, and everyone will have time to leave.
Okay, thank you very much.
So I take it that there are no more questions in the room. Then I have seen 2 questions or 2 hands raised on Zoom. The first one was for Fabrizio. You want to unmute yourself?
I feel like my question was already asked, so I'm good.
Even better then.
I mean, there were so many great questions that I have to say, even I feel like almost all of them have been. I'm curious what Manish will uncover.
Right. I want to know really, so at some point you said that there is a strain-specific threshold for the quorum signal. So finally, in your final model, is that true, or is it simply the dilution of the quorum molecule as the aggregation is occurring that leaves the loners behind? Or is there some kind of a difference in threshold between these different cells?
I'm sorry, I got a little bit, I lost something there. You mean, so here we have 4 parameters that are strain specific: the rate at which they emit the signal, the threshold for the signal, for perceiving the signal, and the rate of stochastic decision making and the velocity.
So all the 4 parameters are strain specific. Sorry, I did not realize that. I thought it was only the sensing threshold that was strain specific.
No, we played with all of them, right? I mean, you can see how they don't influence things independently, right? When you get here, you realize that basically theta is the threshold for perceiving, gamma is the rate of emitting. It's not that they influence things independently, but that there is an important relationship between theta over gamma, and the same with the other 2 rates, lambda over v. So in principle, we didn't have to make all of them different. Maybe not all of them are different, but we did just so we see where the behavior comes from. Yeah.
Well, thank you.
Okay, so it would seem that now we have asked all the questions. So thank you so much.
Completely. Yeah.
So thanks again for your talk.